Okay, think about this for a second. What on earth connects a horse's chronic cough, a dairy cow's fibrotic gland, and a wrinkled, greasy piglet? I know, it sounds like the setup to a really strange agricultural joke, right? But there's no punchline here. The answer actually lies in an invisible world that's all around us, every single day. We are looking at a trio of microscopic villains that are responsible for some of the most persistent, gruesome, and honestly deadly infections in the entire animal kingdom. So, welcome to this explainer. All right, let's dive right into the microscopic rogues gallery of pyogenic and glandular infections. We're going to treat this like a real investigation, opening up the case files on three major bacterial culprits that just wreak absolute havoc on farms, in stables, and sometimes, as you'll see, even in our own lives. Here is our lineup of suspects for today. First up, we have Streptococcus, the pus producers. Then we'll escalate things to Staphylococcus, toxic invaders. And finally, we'll confront Glanders, a fatal threat. But hey, before we meet our first suspect, we really need to understand their calling card. There's a key term that ties a lot of these infections together, and that term is pyogenic. Simply put, pyogenic means involving or relating to the production of pus. Yeah, it's a bit gross, I know, but understanding this specific pathology is absolutely crucial because it perfectly explains the massive, devastating inflammatory response we're about to see in these animals. Okay, moving to section 1, Streptococcus, the pus producers. So specifically, we're tracking an obligate parasite of horses called Streptococcus equi. This pathogen is an absolute nightmare for equine populations. It usually strikes young horses and is almost always brought into a stable by a silent carrier. The disease it causes is famously known as Strangles, or clinically as Adenitis equorum. And the progression is just agonizing. Let's walk through it. Step 1. The horse inhales the bacteria, usually from nasal discharge or a droplet from another horse. Step 2. The pharyngeal and nasal mucosa get infected, which causes acute pharyngitis and rhinitis. Then, by step 3, the infection drains right into the lymph nodes, causing severe abscessation. And finally, step 4, those abscesses spontaneously rupture, releasing large quantities of creamy, yellowish pus. But here's the really scary part. Strangles doesn't always stay localized to the neck and head. Things can escalate dramatically. These abscesses can actually metastasize and spread throughout the entire body, invading the lungs, the liver, kidneys, spleen, and sometimes, incredibly, even the brain. On top of that, those infected lymph nodes can drain inward into the horse's guttural pouches, causing a severe condition called empyema. And if that wasn't enough, the horse can actually develop a sensitivity to the streptococcal protein itself. That triggers purpura hemorrhagica, which results in large areas of edema and hemorrhage under the skin from septic emboli in the blood vessels. It is a cascading disaster. 5. That is the absolute upper limit of days for the incubation period of Strangles. It takes a mere 2 to 5 days, meaning this infection moves incredibly fast. Literally, before a stable manager even realizes there's an outbreak, multiple horses could already be harboring and actively spreading this highly contagious pathogen. Now, horses definitely aren't the only ones in the crosshairs here. This pathogen is super versatile. For instance, in cattle, Streptococcus agalactiaceae enters the teat canal, causing fibroblastic connective tissue to proliferate into a fibrotic gland. That's bovine mastitis, which vets usually diagnose using the California mastitis test. In pigs, Streptococcus porcinus causes cervical lymphadenitis, which is basically swine strangles. And tragically, various strep strains can even enter through the umbilical of newborn mammals, including foals, calves, and human infants, resulting in a suppurative polyarthritis that we call jointill or navel ill. All right, let's move on to our second suspect in section 2, Staphylococcus, toxic invaders. We are seriously leveling up here. We're moving from simple pus producers to heavily armed toxic invaders like Staphylococcus aureus, Staphylococcus epidermidis, Staphylococcus intermedius, and Staphylococcus haeicus. What's mind-blowing here is just the sheer scale of the pathogen's arsenal. I mean, look at the weapons it brings to the fight. It uses surface proteins just to adhere to the host. It has specific receptors for fibrinogen, fibronectin, and vitonectin to literally anchor itself to endothelial cells. And it actively dodges the host's immune system using protein A, which binds to Fc proteins to shut down chemotaxis and phagocytosis. Toss in protein-degrading enzymes and a brutal cocktail of toxins, coagulases, fibrinolysins, hyaluronidase,
4 different types of hemolysins, plus enterotoxins, and you're looking at a microscopic war machine.
So what does all this weaponry actually do? Well, it leads to a chronic pyogranulomatous inflammation known as botryomycosis, and this hits cattle, sheep, horses, dogs, cats, and pigs. On a gross level, you'll see these lesions in the subcutaneous tissues as multiple firm nodules, all with purulent centers. People sometimes call these purulent grains sulfur granules. Under a microscope, it's just a massive cluster of neutrophils surrounded by granulation tissue. The clinical result? Fleshy nodular growths that vividly and rather gruesomely resemble a bunch of grapes. Another major staphylococcal threat is exudative epidermitis, caused by Staphylococcus hyacus. And hey, this is the answer to our greasy piglet riddle from the very beginning. Commonly known as greasy pig disease, this specifically targets suckling piglets. Now, S. hyacus is actually just a normal inhabitant of a pig's skin, but when it turns pathogenic, it triggers generalized seborrheic dermatitis. In its paracute form, the poor piglet suffers from cutaneous erythria. Their entire skin coat becomes wrinkled, reddened, and completely covered in a greasy gray-brown exudate because the bacteria's lipase literally destroys the protective lipids right on the skin surface. Okay, this brings us to Section 3, Glanders, a fatal threat. We are now at the absolute climax of this explainer. We've seen the pus, we've seen the toxic war machines, but now we have to introduce the single most dangerous suspect in our lineup, glanders. It's caused by Pseudomonas malai, which you might also know by its older name, Pfeifferella malai. This is a gram-negative obligate parasite, and it causes a highly contagious, highly fatal disease in horses, mules, and donkeys. Now, unlike Strangles, which is inhaled, glanders mostly spreads by ingestion. The bacteria contaminate fodder and watering troughs through infected nasal discharge or sputum. Once an animal eats or drinks it, the bacteria invade straight through the intestinal wall, eventually leading to a terrible death by anoxic anoxia. Basically, the body's tissues are completely starved of oxygen. And what's really wild is how this single disease impacts different animals in completely different ways. If a donkey ingests Pseudomonas malai, it almost always develops the acute form. High fever, septicemia, rapid onset of severe signs, and death within a matter of days. But if a horse gets it, they tend to develop the chronic form, bacteremia, where the bacteria actually localize in the lungs. This causes a persistent chronic cough, frequent nosebleeds, and really labored breathing. Vastly different timelines, but both have equally tragic outcomes. Glanders physically manifest on the body in 3 horrifying ways. First, you have nasal lesions. These are deep, erosive ulcerations right over the mucosa, especially the septum, and they come with a copious, blood-tinged, purulent discharge. Second are the pulmonary lesions. These are discrete granulomatous nodules that look a lot like tubercles. Inside these granulomas is a caseous, or cheese-like, necrotic center, all surrounded by epithelioid cells, giant cells, and lymphocytes. And third is cutaneous involvement, which is known as farcy. This produces indolent, meaning painless, persistent ulcers on the skin, mostly on the legs, and they're connected by these tortuous, heavily thickened superficial lymphatics. Now, if an animal somehow manages to survive a localized nasal lesion long enough for it to actually heal, it leaves behind a very specific physical brand. The healed ulcer on the nasal mucosa forms a characteristic stellate or star-shaped scar. It is a permanent, absolutely unmistakable footprint of Pseudomonas malai. Historically, figuring out if an animal had glanders involved a pretty grim procedure called the Strauss test. Basically, they take infected material and give an intraperitoneal injection to a male guinea pig. The infection was so terrifyingly fast that by day 3 or 4, acute purulent orchitis would develop, confirming the bacteria was present. Thankfully, we don't have to do that anymore. Modern veterinary medicine relies on the intradermal Malin test and DOT ELISA tests to quickly and safely spot outbreaks before they spread. But here is the absolute most critical takeaway. Glanders is not just an agricultural problem. It is a massive zoonotic danger. Humans are highly susceptible to Pseudomonas malai. If a farmhand or a stable manager or a veterinarian accidentally comes into contact with this pathogen, the resulting infection is usually fatal. That elevates glanders from just an agricultural nuisance to a literal top-tier biological hazard. Which brings us to our final thought for today. We've investigated the pus-producing streptococci, the wildly toxic staphylococci, and the very real fatal threat of glanders. And honestly, it really makes you wonder, if a microscopic pathogen can jump from a simple farm watering trough and cause a completely fatal human infection, just how secure is that invisible border between the barnyard and our own lives? Definitely something to keep in mind the next time you're visiting a stable. Thanks so much for joining me on this explainer, and keep questioning the microscopic world around you.